Okay, picture this. Imagine having the power to actually edit the very code of life, snipping and pasting DNA with absolute pinpoint precision. Now, what's truly mind-blowing is that the tools we use to do this, they weren't cooked up from scratch in some modern chemistry lab. They evolved naturally. Today, in this explainer, we're diving into restriction enzymes. These are nature's own microscopic scissors, originally developed by bacteria just to chop up invading viruses. We're going to break down exactly how they work and how they absolutely revolutionized biology as we know it.
So here's our roadmap for this explainer. First up, we'll meet the molecular scissors. Second, we'll look at finding the target sequence. Third, making the cut. And finally, building with sticky ends. Let's get right into it.
Section 1. Meet the molecular scissors. Now, in molecular biology, we literally call these enzymes scissors. But out in the wild, in nature, they're found inside bacteria. And the bacteria use them as a built-in defense mechanism, basically a weapon to kill viruses. When a virus attacks and injects its genetic material, the bacteria deploy these enzymes to aggressively attack that viral DNA, just chopping it up and breaking it into totally useless fragments. It is this incredibly ruthless microscopic defense system that humanity has brilliantly co-opted for our own use.
Getting down to the chemistry of it all, what exactly are these things? Well, the formal scientific term for our molecular scissors is restriction endonucleases. Basically, these are enzymes that recognize highly specific DNA nucleotide sequences, and then they do something pretty amazing. They cleave or cut the DNA double helix right at or very close to those specific sites. And it's exactly that ability to cleave the double helix predictably and reliably that makes them such a powerful tool.
But there isn't just one single type of molecular scissor out there. You've actually got 4 main flavors of endonucleases. Type I recognizes a sequence but cuts the DNA randomly, sometimes up to 1,000 base pairs away from the recognized site. Type 3 cuts somewhere within 25 base pairs. Type 4 cleaves only modified DNA, like methylated bases. But honestly, scratch those for a second, because I really want to highlight Type 2. Type 2 enzymes recognize specific sequences and cleave at constant, hyper-precise positions right at that exact sequence. Because they don't guess and they don't cut randomly, Type 2 enzymes are the absolute stars of the show. They are, for sure, the most useful type when it comes to genetic manipulation and cloning.
Which brings us to Section 2, finding the target sequence.
So, a single strand of DNA is incredibly vast, right? It's massively long. To find their specific cutting site, restriction enzymes have to literally physically scan this DNA code. They just glide along the molecule, hunting for a very specific set of nucleotides. And usually the target sequence they're looking for is surprisingly short, only about 4 to 8 base pairs long. It truly is like finding a microscopic needle in a massive genetic haystack.
The restriction enzymes that we use most heavily in gene manipulation are looking for something super specific. Short palindromic sequences that have an axis of symmetry. Think about it like this. If you look at the top strand of the DNA, reading from the 5' to the 3' direction, you might see a sequence like TTAGCAC. Now, if you check the bottom strand directly below it, reading in that exact same 5' to 3' direction, it's the exact same sequence, TTAGCAC. It reads the exact same forwards on the top as it does backwards on the bottom strand. That perfect structural symmetry, that's exactly what the enzyme is designed to recognize.
Now, the names for these restriction enzymes might look a bit like alphabet soup at first glance, but they actually tell you exactly where the enzyme comes from. They're named for the producing organism and the order they were isolated. For example, EcoRI was the very first restriction enzyme isolated from the Escherichia coli strain RY13. Or HIND3, that was the third enzyme isolated from Haemophilus influenza strain ARD. And the really cool part is that different bacteria produce entirely unique scissors tailored for completely unique codes. EcoRI is hunting for GAATTC, while HAY3 is looking for a much shorter GGCC. By mapping all of this out, researchers have built up a massive catalog of different biological scissors to choose from.
Moving right along to Section 3, making the cut.
So what actually happens when the enzyme cleaves the DNA? We generally see 2 main types of cuts. An enzyme like HAY3 will cut straight down through both strands. That creates a really clean, flat edge, which we call blunt ends. But then you have an enzyme like EcoR. It recognizes its specific site and cuts in a very specific, jagged pattern. This produces a staggered edge, leaving single-stranded DNA overhangs totally exposed.
There's a line directly from our source material that I absolutely have to highlight here. EcoRO1 makes a staggered cut and produces a sticky end. That phrase right there, sticky end, is a literal game changer in biology. See, while blunt ends are just two flat edges that kind of bump into each other, those single-stranded overhangs from a staggered cut naturally want to pair back up with their matching sequence. Because they are actively seeking out complementary base pairs, they're chemically sticky. And this stickiness is the very thing that allows us to hack life's code.
And that sets us up perfectly for Section 4, building with sticky ends. Okay, here is the sheer magic of molecular cloning. Imagine we have two completely separate pieces of DNA. Let's say a target gene we want to isolate and a circular piece of DNA called a plasmid. We expose both of them to the exact same molecular scissors, EcoRI. Because EcoRI is searching for the exact same GAATTC sequence everywhere it goes, it makes the precise same staggered cuts in both the target gene and the plasmid. After the cut is made, both pieces have perfectly matching sticky ends. The single-stranded overhangs on our target gene are perfectly complementary to the overhangs opened up in the plasmid. We've effectively created matching biological puzzle pieces.
So here is that physical cut and paste action laid out 1, 2, 3. Step 1, our enzyme, EcoRI, creates complementary sticky ends on DNA from two entirely different sources. Step 2, because those sticky ends are complementary, they naturally want to bond. They literally hold the two pieces of DNA together temporarily. But they aren't fully secure just yet. They need a permanent fix. And that brings us to Step 3. Another enzyme called DNA ligase enters the chat. If restriction enzymes are the scissors, DNA ligase is the glue. It links the DNA backbones permanently together, finalizing a brand new combined DNA molecule.
So, why does all of this matter? What's the big takeaway from this explainer? Well, the power of these molecular scissors extends far, far beyond a microscopic bacterial battlefield. By allowing us to combine DNA sequences from entirely different sources, restriction enzymes gave us the ability to construct physical restriction maps. They allowed researchers to sequence massive genomes. They're the exact tools we use to build vast gene libraries. Ultimately, these enzymes are the foundational tools enabling all of molecular cloning. Literally every genetic advancement in recent decades starts with these tiny molecular scissors. It's the whole shebang.
We've explored how bacteria fought viruses and how science borrowed their ancient weapons to literally read and write the code of life itself, but science never stops moving forward, does it? Now that we actually hold the microscopic scissors of life and we've mastered the intricate art of cutting and pasting genes with sticky ends, what new molecular masterpieces will we build next? It is a truly fascinating frontier. Thank you so much for joining me for this explainer. Keep exploring, stay curious, and I'll catch you next time.